Hi, everyone. Thank you for coming to listen to the Caribou story. So over the next few minutes, I'm going to tell you a little bit about the company and who we are and a bit of our story. Two, I'm going to tell you a little bit about our platform technology, and then I'm going to tell you a little bit about our lead product, CB10. And then if time permits at the end, I'll just touch a little bit on our Microbiome Platform. So to start, let me tell you a little bit about Caribou as a company. We were founded in 2011 with technology, CRISPR-Cas9 technology, coming out of the lab of Jennifer Doudna at UC Berkeley. And initially, Caribou was very much a platform company. And at that time, we decided to work with various partners to exploit CRISPR in as many areas as possible. During that time, Caribou as a company, we continued to work on gene editing platform with the idea of improving the technology. And eventually, we came up with a second-generation technology that we call Chardonnay, and I'm going to tell you about it, that actually has greater specificity than the first-generation technology. With that second generation, we've been able to pivot from a platform company to a products company, and we've been building a portfolio of therapeutics. And we're doing that in two areas. The main area where we work is allogeneic CAR T-cell therapies, and I'll tell you about our programs. The second area where we're also active is we use gene editing to edit gut microbes. We've raised $41.5 million so far. We have about 65 employees. This slide shows you the management team. So the team at Caribou is a nice mix of people who were pioneer in the gene editing space. Our CEO, Rachel Harwitz, came from the lab of Jennifer Doudna, as well as people like myself, Steve Kenner, our CSO, Barbara McClung, our CLO, who have a long experience both in the pharma and the biotech industry. This year, we've also expanded our scientific advisory board that really now reflects the efforts of the company. So on the SAB, we also have a mix of people with the gene editing experience, so Jennifer Doudna is still on the board, as well as people that have experience with CAR T-cells. So we have people like Cameron Turtle or Nupur Raj. And then we also have now a few people that are involved in the microbiome space, like Emily Boscos or Amy Bott. I probably don't need to tell this audience about why we're interested in allogenic therapy and why we believe that next-generation therapies will be allogenic and no longer autologous. As you know, there's great efficacies with the autologous therapy, but there are also significant limitations from vein-to-vein time to the manufacturing challenges. An allogenic approach gives you the ability to have an off-the-shelf product. It also gives you the ability to start from T cells from a healthy donor. So hopefully a better product and a more consistent product. And it obviously gives you manufacturing advantages. This slide shows you our pipeline of products. As I mentioned, we're focused in two areas. The first and the primary area at this stage is ex vivo immune cell therapies. Our lead product is CB10. It's a CD19-directed CAR T-cell where we do a couple of edits. We insert the CAR into the T-cell receptor locus, and we also knock out PDCD1, which is the gene that encodes for PD-1. And I'll tell you a bit more about CB10 in a few slides. Our second product is CB11. It's also a CAR T-cell. We haven't disclosed the molecular target. And in the case of CB11, we actually do slightly more sophisticated editing. For CB11, we do 2 knock-ins. And then we have CB20, which is another immune cell, and then CB31 will be our first product in the gut microbe area. So let me take a few slides to tell you about Chardonnay, our second-generation gene editing technology. For those of you who are not familiar with CRISPR, at the heart, it's really the combination of two things. It's the combination of a nuclease, which typically is a guide made of RNA, and an enzyme, which in our case is Cas9, and the two form a complex that will actually recognize a particular site on the DNA and do a double-stranded cut. This cut can be replaced through a couple of pathways, resulting either in knock-ins or knockouts. As most of you know, it's a very versatile technology. It's heavily used, but one of the limitations is that in addition to cuts in the intended sites, you can also get off-target effects. And so what we've been doing at Caribou is working on a second-generation technology where we leave the nuclease as is, so we continue to work with Cas9, but we try to engineer the guide so that it's no longer made just of RNA. And what we've developed are guides that are made of both RNA and DNA, and we call those CRISPR hybrid RNA DNA, or Chardonnay for short. And by doing that, we actually modulate the affinity of the guide for the target sequence and can get a higher specificity. And I'll show you that higher specificity in a few slides. The other advantage of the Chardonnay technology is that it's got brand new IP. So let me show you a little bit how robust the technology is. And here on this slide, we're actually looking at editing efficiency. And we're looking at a variety of genes. So each different color is a different gene. And for each gene, we're actually comparing a traditional RNA guide in the darker color versus a Chardonnay guide in the lighter color. And we actually look at various loci on the gene. And what you can see is that routinely, no matter what gene we look at, we can routinely find Chardonnays that have the same editing efficiency as a traditional RNA guide. This slide shows you the advantage we get on the specificity side. So here what we're doing is we're looking at off-target effects for a variety of genes. And again, we're comparing the traditional RNA guide versus a Chardonnay guide. And so for example, if we take the example of TRAC, the T-cell receptor alpha constant locus in red, we're comparing, if we look at the on-target effects, both the RNA and Chardonnay have very similar on-target effects. But the old RNA guide will have a few off-target effects that, at the limit of detection, we don't see with our Chardonnay. And it's true across genes and across sites on gene, even for AAVS1, which is a gene that's well known in the gene editing community as very promiscuous, where, in this case, the old RNA guide gives a fair amount of off-target effects. We don't see anything with Chardonnay. The other thing we've been focusing on as we develop the platform technology is translocation. So as soon as you start to do two edits, as soon as you start to do multiplex editing, there's a concern for genomic translocation. And this is something that has been reported by others. Selectis has published a paper where, using their technology, they see about 3 to 5% translocation. And so what we have in our case is there's two things that are helping us to minimize translocation. The first thing is the fact that the Chardonnays that we're using have much less off-target effects. So that, in effect, ensures that whenever we use the technology, we do get a single cut. The other thing we've done is we've developed a proprietary protocol in the way we administer the reagent so that we actually minimize translocation. And if we compare the traditional way of delivering the reagents versus our proprietary delivery, we go from seeing 3.5% translocation to barely any. So overall, we've developed a very robust platform that allows us to do gene editing in a very precise fashion, much less off-target effect, much better control of translocation. And so we've been using that to make our therapeutics, and I'll tell you now about CB10, which is our first product. So as I mentioned, CB10 is a CD19 CAR T cell where we make a couple of edits. The first edit that we make is that we selectively insert the CAR into the T-cell receptor so that in a single step, we knock out the T-cell receptor, which is critical to avoid graft versus host disease, but we also insert the CAR into a well-defined location in the genome. The other edit that we make is that we knock out PDCD1, which is the gene that encodes for PD1, and by that, we hope to get greater persistence of the T cell. This slide shows you a couple of examples of papers from the academic community looking at manipulating PD1 to increase the potency of CAR T cell. The example on the left is a paper from Alex Marson's lab at UCSF, where pretty much he does what we're doing, which is he creates CAR T cells that also have PD1 knocked out, and he shows in animal models that you actually do see greater anti-cancer effect. The example on the right is a paper from Rainier Bretchens at Memorial Sloan Kettering, where in his case, he actually creates CAR T cells that secrete an scFv to PD1. And again, he sees that this allows to get greater anti-cancer effect. So this slide shows you a bit of in vitro data on our own CB10 product. On this slide, we're looking at two different models. On the left, it's an ALL model. On the right, it's a DLBCL model. And we're comparing in red the CB10 CAR T slides versus in blue control slides that don't have the CAR, just have the T-cell receptor knocked out. And as you can see, when we look at lysis of cancer cells, we actually have a very nice dose-dependent killing of the cancer cells with the CAR T that corresponds to secretion of interferon gamma. This slide shows you in vivo data. So in this case, we actually use a xenograft model where we orthotopically implant an ALL model. This is a cancer that expresses CD19 as well as PD-L1. And we actually treat the mice either with CB10 in red, a T cell that has the CAR but that doesn't have the PD-1 knockout in green, and then a control cell in gray that has the TCR knockout but no CAR. And as you can see, we get good anti-tumor effects with just having the CD19 CAR in green, but the deletion of a PD-1 in red gives us even greater longevity and anti-tumor efficacy. Finally, let me just say 2 words about our Microbiome Platform. So in the microbiome area, we have a very different approach than the rest of the field. If you look at companies working in this space now, they tend to fall into 2 buckets. The first one are companies that manipulate consortia of existing microbes. The second one are people who try to engineer the microbes, but typically the people who try to engineer the microbes are limited to microbes for which there's a variety of tools that have been developed, such as E. coli. What we're trying to do is have a broad platform using CRISPR gene editing so that we can actually engineer any gut microbe. And so in order to do that, we've developed a variety of tools. The first thing we've developed internally is the ability to identify, isolate, and characterize a variety of strains. And initially, our interest is really around Bacteroides lactobacillus. We've also been working on having a technology to actually deliver the CRISPR materials to the bugs. As you may know, electroporation will sometimes work with the bugs, but it's not a very general approach. So we've developed an internal phage delivery technology to deliver the CRISPR materials. When you want to use CRISPR in microbes, it's very different than in eukaryotes. I've shown you how it works in eukaryotes. You get a double-stranded cut that can be repaired in a variety of ways. In microbes, a double-stranded cut will actually lead to a death of the microbes. And so what we've developed are alternative variants of the technology. So we use, for example, nicases, which allow you to do a single-stranded break. We also use dead-Cas9, which gives you the ability to not do the double-stranded cut, but actually modulate gene expression. And so these are the tools that we've developed to engineer gut microbes. And then finally, we've developed in vivo models so that we can actually look at both the PD and the PK effects of our gut microbes. So as a conclusion, I hope I've given you a nice overview, first of our 2nd-generation gene editing technology, Chardonnay. I've shown you that it really improves specificity over the 1st generation. I've also told you a little bit about the portfolio of products we're building. I told you about CB10, our first product, which is a CD19 allogenic CAR. And then I've given you a little bit of a flavor for what our microbiome platform is. I have about a minute left. I'd be happy to answer any question. Otherwise, I'll be outside and happy to take any questions there. Thank you.